All right, everybody see that once in a lifetime opportunity slide? So I'm extremely grateful to Mike for inviting me to speak to you because ever since this work of Frederick Johansson that I'll be featuring in this talk came to my desk now 7 years ago, I've been on a crusade to try to persuade the molecular biology community that there's a huge opportunity staring them in the face, um, that they can actually find the engram. And finding the engram will be as important to neuroscience as Avery and Watson and Crick were to biology. Um, so this the work at the core of what I'm talking about was done by Frederick Johansson, who at the time was a doctoral student in the lab of Jerry Heslow at Lund. For those of you who don't know, I certainly didn't know, Lund is the largest university in Northern Europe. Anyway, Frederick now has his own lab in the medical school there at Lund. And Jerry, who's closer to my age, although considerably younger than I am, has spent his entire career working on the neurobiology of the condition eye blink reflex, which has long been recognized by people interested in finding the engram as perhaps the most promising behavioral preparation. And so you need to know something about the electrophysiology, the neuroanatomy, and the conceptual background here. But the star of this show is the Purkinje cell. I'm sure many of you already know about it, but in case you don't, it's a really spectacular neuron. It's pretty clearly one of the most spectacular neurons in the entire brain. And here's a wonderful picture of it off the internet. And as you can see, it's this huge neuron, one of the largest neurons in the brain, with this enormous flat. It's not obvious from this photograph, but this dendritic tree is flat. This is essentially a two-dimensional structure, right? It's a chip on a bus. If ever there was one, a neurobiological chip on a huge data bus. And the anatomy of the system is very well known. This neuron gets, as is most of the pharmacology, it gets two excitatory inputs, one of which comes from the parallel fibers. And the purpose of this dendritic tree is to enable up to 200,000 different parallel fibers to synapse on this dendritic tree. And those parallel fibers bring signals from every modality and from all over the body. And the other input to this cell is a single fiber. You can see a few of them here. The climbing fiber that comes up from the cerebellar nucleus, acerebellar nucleus. No, I'm sorry, a deep midbrain nucleus. There are also three inhibitory interneurons. And another thing you need to know is that the axon from the Purkinje cell is the sole output of the cerebellar cortex. So the particular Purkinje cells that Johansson was working with are in one of the very small microzones of the cerebellum. And the cerebellum has zones. The stripes here are examples of the zones. And then within those zones, it has microzones. And the microzones are defined by the part of the animal's anatomy from which the climbing fiber projects. So remember, each Purkinje cell only has one climbing fiber input. And the climbing fiber inputs also come from all over the body, but each microzone is only the right way to think about a microzone is it's looking at the extent to which signals from all over the body and every modality can predict events that occur in a very localized part of the body. All right. So the parallel fibers bring in the signals from all over, and the climbing fiber brings the information from a highly localized part. And this microzone is devoted to the skin around the eyes. And just to give you a sense of why they call it microzone, that little black, this is a top-down view of the cerebellar cortex of the cat, and that little spot, that's the microzone in which Frederick Johansson did the experiment. So another thing you need to know, and again, I understand, I'm no little doubt that many of you, perhaps all of you, know this, but still I want to make sure we get all the background in place. The electrophysiology of the cerebellum is, of the Purkinje cell in particular, is fascinating because the two different inputs produce two different spikes. If you drive the parallel fiber input to the Purkinje cell, under the right circumstances, not all circumstances, you will see spike per spike driving. That is, each input spike drives a simple spike. And these simple spikes, as the name implies, are the kind of spikes we're all familiar with, right? Garden variety, Hodgkin-Huxley spikes. The Purkinje cell fires at a very high endogenous rate. So, which is endogenously generated spontaneous activity in the 40 to 100 hertz range. So these spikes that are driven by parallel fiber input are intermixed with the endogenously generated spikes. And the synaptic input from the parallel fibers is glutaminergic. So the climbing fiber, by the way, the climbing fiber, when it reaches the Purkinje cell, branches all over the place. It has a huge axonal arbor that sort of embraces the Purkinje cell and the trunks of its dendrites. And that synapse is also glutaminergic, but bursts, high frequency bursts of spikes in the climbing fiber input produce complex spikes in the Purkinje cell. And they're called complex spikes because they reverberate. You don't just get your usual one spike, you get this kind of thing. And it can go on for quite a while, as this illustrates, right? And as far as I know, no one really understands the mechanism that underlies these complex spikes. And that's worth keeping in mind, because in pursuit of the project that I'm trying to persuade you to pursue, one of the byproducts would probably be an understanding of what the hell's going on with these complex spikes. Okay, now turning to the behavior. And again, I have no doubt many of you know this. This is right from your introductory psychology course. So Pavlovian conditioning is this basic form of associative learning pioneered by Pavlov, in which there's a neutral stimulus called conditional or conditions, one or the other stimulus, which warns of an impending event. For Pavlov, it was a bell or a touch to the skin. He actually used lots of different CSs. And the impending event was the delivery of food, the intraoral delivery of food, which elicited salivation. He had won the Nobel Prize for studying the physiology and neurobiology of salivation, which is the So the food was the unconditioned stimulus and salivation was the unconditioned response. But when the same response or a related response, there's a whole story there itself, is elicited by the CS rather than by the US, then it's called a conditioned response. It's an anticipatory response to the CS because the animal has learned that the CS predicts the US. Now, enormously important and often glided over in introductory psychology, not to mention in neuroscience textbooks, is the fact that in all Pavlovian paradigms, the conditional response is appropriately timed. It's awkward because the standard neuroscience story about memory has nothing to offer in the way of an explanation about where the timing comes from. In other words, in the course of a necessary precondition for the development of this response is that the animal learn the interval that between the CS and the US. And in eye blink conditioning, you can vary this. Actually, probably you can vary it over a very large interval, but in the experimental literature, it's been varied from less than 100 milliseconds you cannot condition the preparation, basically because there isn't time to make the conditioned response, the anticipatory response. And the interval can extend out to 2 seconds. It could almost certainly extend much longer, but for various reasons, no one's investigated anything beyond 2 seconds, at least not that I know of. Now, a hugely important fact that was discovered in the 1990s, if I recall. The focus of the neurobiology of eye blink conditioning was on, guess what? The hippocampus for decades, until 2 labs independently looked for it in the decerebrate preparation. And I think you all know the decerebrate, there is nothing above the midbrain. And lo and behold, you get lovely eye blink conditioning in the decerebrate preparation. So that greatly simplifies things. We can forget about the forebrain. Okay, now we come a bit to the conceptual background you need. So the first, I'm not going to dwell on this, this is a whole separate talk, but if you've been taught anything about associative learning, you have to forget what you've been taught because it's all rubbish. So it's all about the formation of associative bonds, and you can trust me, in 50 or 100 years when the history of psychology, neuroscience is written retrospectively, the associative bond will be regarded as the phlogiston, the philosopher's stone, the luminiferous ether of psychology. And plastic synapses are just a neurobiological euphemism for the material realization of the associative bond. So what you need to know about associative learning is that the animals learn the facts. They learn all the intervals in the protocol, not just the CS-US interval. They learn the US-US interval. In fact, they learn the mean and variance of all these intervals. They learn the CS-CS interval, which is not necessarily the same as either of these intervals, and they learn the session duration. They probably, in fact, they also learn how long they've been being run in this experiment, which is an interval, which is usually measured in days or even months. Now, what's important for this program that I'm trying to persuade the molecular biology community to embark on is that remembered intervals are maximally simple empirical facts, right? If you say, well, the animal learns facts, people say, well, facts, whoa, what's a fact? Most of the facts that we carry around are very complicated things, right? What is it? I mean, it's a fact that Trump is no longer president, right? But yeah, that's a complicated fact. And I mean, it's so complicated that 40% of the population doesn't believe it. So if we're going to look for the mechanism that stores facts, and that's what we're looking for when we look for the engram, we need really simple facts and ones that are very easily manipulated empirically. The durations of intervals are exactly that. And another point to bear in mind is that when we talk about a quantitative fact, like the duration of an interval, and we talk about how it's represented, we're really talking about numbers. I mean, numbers are the symbols that we use to represent quantitative facts. So what you're really looking for are numbers in the physical realization of numbers in the brain. If you like, you're looking for a brain numeral. That's to distinguish between the number, a qua Platonic concept, and the actual physical realization. Okay. So memory is a repository of facts gleaned from experience. Its function in the brain's computations is to carry forward in time the facts, the quantitative facts. In the end, all facts are quantitative. For use in computations that direct subsequent behavior. For example, as I talk, I'm using the words of English, and most of them have been kicking around in my brain now for close to 80 years. So what memory does is make possible the temporally unbounded composition of functions. Computation is the composition of functions. That is, you perform one function, you add a couple of numbers, you store the result, and then later on you may take that stored result and use it to multiply some other number. That's the composition of functions, feeding the output of previous functions to the inputs of new functions. And that's what basically computation is all about. Now, that presupposes, I mean, if the brain really is a computational organ, and that's the foundation of my philosophy, that presupposes that the facts, the numbers, have been encoded in some manipulable form. That is, they can be processed by addition, multiplication, and so on. So what are we looking for when we look for the engram? We're looking for the material that encodes the facts. And I already told you how important this is. Okay, what Frederick showed is that the Purkinje cell times the conditioned response. Actually, that was already known, right? So in this long history of the neurobiology of the conditioned eye blink, one of the huge discoveries was the discovery that it's intact in the decerebrate preparation, because that completely refocused the locus of neurobiological inquiry. And then the question was, well, what is it in the midbrain that's important? And as usual, there was a lengthy controversy, but now there's pretty general consensus that it's the cerebellum, and more particularly the cerebellar cortex, and most particularly the Purkinje cell. Because what isn't in dispute, and this is not really very much in dispute, what I just said, these disputes linger forever. What isn't in dispute is that in the course of conditioning, the Purkinje cell in that little microzone develops a learned pause in its spontaneous firing rate. So this cell is there firing 80 hertz very irregularly, by the way. The distribution of interspike intervals in the Purkinje cell is really weird. But sorry, let me eliminate this. What isn't in dispute, because it's been repeatedly confirmed in many different labs, is that in the course of developing the conditioned reflex, when the animal's being trained, a pause appears in this conditioned firing rate, and the pause is proportional to in fact, roughly equals, the duration of the pause roughly equals the CS-US interval. Moreover, the output of the cerebellum, as I think I already said, the axon is the only, I'm in the midst of my Zoom talk. Okay. Okay. What's not in dispute? The output of the Purkinje cell is the only output of the cortex, and it's an inhibitory. So when it pauses, it's releasing an inhibition on cerebellar nuclei, which is so it almost certainly controls the duration of the latency of the eye blink. Now, some work of my own using the data from that Frederick produced shows another curious property of this system, which sooner or later is going to be important in finding the engram and understanding how it works, is that so the pause has an onset, right? And it has an offset, right? And the onset and offset are stochastically independent. Later on, I'll show you an actual data and you can see that they're stochastically independent. What that means is that they're both proportional to the CS-US interval. So they're both accessing the engram. And they both show variability. In fact, scalar variability, which is a signature of conditioned responses. But on a trial in which the onset of the pause is unusually long, that doesn't predict that the offset of the pause will also be unusually long. So this is important in that whatever the pathway is from the engram to the output, it has two independent sources of noise. In some sense, the engram is read twice, once to control the onset of the pause and once to control the offset of the pause. Okay, so now we come to the essence of Friedrich's experiment. So here we have a Purkinje cell, and Frederick put a stimulating electrode on the parallel fibers. Right, here's the data bus. And he put another stimulating electrode on the climbing fiber coming from the olivary nucleus, and a recording electrode up against the cell body of a Purkinje cell. And then he trained that Purkinje cell. That is, he used the stimulation of the parallel fibers to generate the CS signal. So he had control over the data on the data bus. All right, and that's incredibly important in understanding why this experiment is so important. So his CS, this is the object of sort of neurobiology, right? To get rid of the behavior, to simplify it down to here. And now we're just working at the electrophysiological level. The CS is the stimulation of the parallel fibers. The US is stimulation with two quick bursts of very high frequency pulses of the climbing fiber to produce a complex spike. And in different preparations, different decerebrate ferrets, he works with the ferret, he varied the CS-US interval, the latency at which the onset of the CS stimulation predicted the onset of the US stimulation. Now, another lovely part of this experiment was that, contrary to the usual behavioral practice, where the US occurs at the moment when the CS ceases, he wanted to know whether the offset of the pause was a response to the US or a response to the CS. So he continued his CS stimulation for 800 milliseconds, even though the range of CS-US intervals that he used was from 150 milliseconds to 400 milliseconds, right? So in his experiments, the CS input continued for hundreds of milliseconds after the time at which the US was anticipated. So, oops, sorry. Yeah, so I've already said all this. Got a little ahead of myself here. Okay, here's some of Frederick's data. This comes, I think this particular record was actually published when they published it in the Proceedings of the National Academy. So this, after the conditioned pause appeared, right, then Frederick ran a sequence of 20 probe trials during which there was no US. Okay, so one of the really striking things here, I'm just showing you the no US probe trials, but they look exactly like the US, the training trials in which the US was already present. Whether the US persisted beyond the anticipated time, right? Here's the onset of the CS. Here's the time at which the US is anticipated. I developed an algorithm that identified where the pause began and where it ended in a record like this. That if it's a no-brainer, I mean, it's pretty obvious, but in some of the records, it's not quite so obvious. And so these are algorithmically determined estimates of the pause onsets and pause offsets. And notice I called attention to the fact that they were stochastically independent. So here you see a very long onset and an unusually short offset. Right? So that's what stochastically independent means. And if you look, you can see that this does not predict that with any fidelity. Okay, over here are the distributions of the intervals between the spikes. Each dot in, oh, I realize some of you may not be electrical. Okay, this is a raster plot of each line is a trial. The dots are the spontaneous spikes in the firing of the Purkinje cell. There are 20 of these trials. Sorry, I realize I got a bit ahead of myself here. Okay, so one can look at the distribution of inner spike intervals before the pause. During the pause, in this unusually clean record, there are essentially zero inner spike intervals within the pause, but often there are one or two. Well, I regret saying what I just said. I don't want to go there. But this is the distribution before. This is the distribution after. You can see they look very the same. What's the distribution between the blue lines here, right? Within the quote pause. Well, you can see it looks just like these, except for out here, except for the fact that you have on these unusually long intervals. And in this record, it's obvious that it's basically a single interval. Now, notice that you do have long intervals outside that too, right? I mean, they occur within, it's a very irregular firing, and you can get pauses that are just as long or longer outside the learned ones. Okay. So here's the crucial results that in the unconditioned cell, when you first start training, the Purkinje cell may very well respond to the CS, the stimulation of the parallel fibers, with spikes intermixed among the spontaneous spikes that are obviously coming from the data bus, from the parallel fibers. This is not true in all preparations. Sometimes there's no sign of these spikes. This is not even remotely surprising because it's a glutaminergic synapse. So it's notionally an excitatory synapse. So that's more or less what you expect. What you don't expect is the condition pause after you have trained the cell, and the number of trials that it requires before the pause appears, which is one of the things I analyzed, varies hugely. Sometimes it appears immediately by the second trial. Sometimes it takes hundreds of trials. The same thing is true at the behavioral level. In some animals, the condition blink appears almost immediately, and others it takes hundreds of trials. So the behavior of this cell mirrors at the quantitative level everything we know about eye blink conditioning. And in the whole huge literature on the neurobiology of learning, this is the only thing of which that can be said. If you look in the LTP literature, nothing you find at the cellular level matches anything you find at the behavioral level when you get down to the quantitative facts. Now, the pause lasts only as long as the CS-US interval, regardless of how long the CS stimulation lasts. So what that tells you is that it's only the onset of the firing in the data bus that triggers the reading of the engram. And then from there on in, what the remaining stuff that's coming in on the input is irrelevant. And Frederick actually did lovely variations on that. So he trained with 800 millisecond stimulus, CS stimuli, 50 pulses per second. But during the probe trials in some preparations, he used CSs that were as short, the total duration of the CS was 17 milliseconds. And it was at 500 hertz. The pauses that he got looked exactly the same, whether he used that as the CS input or an 800 millisecond at 50 hertz input. So that's very strong evidence that it's just the first spike, the first spike volley that matters. And everything else from there runs its course regardless of what else is coming in. Okay, so I just said that. Okay. Now, the very important pharmacological results. So the fact that you get a pause, an inhibition of the spontaneous firing rate is itself a big puzzle because the input is glutamatergic. So your first thought is, well, it must be due to that stimulation must somehow have driven one or more of those inhibitory interneurons. But Frederick showed that gabazine in doses, well, and there is a profound inhibition that can be demonstrated in this preparation. If you move your stimulating electrode, the CS electrode, so that it's off beam, so that it's stimulating parallel fibers that synapse on other Purkinje cells, then that stimulation produces a profound inhibition of the firing, the spontaneous firing of the one you're recording from, the Purkinje cell you're recording from. Frederick put in gabazine in doses that completely blocked that effect. And they had no effect on the triggering of the conditioned pause. Right? So that makes it extremely unlikely that the pause is actually traceable to those inhibitory interneurons. And the next thing he did, the second thing here, makes it, as far as I'm concerned, is the final nail in the coffin. Of the interneuron hypothesis, he went on to show that if there are 7 different postsynaptic receptor molecules in this synapse, all of which, to all of which glutamate binds, right? So this is not your ordinary everyday textbook, one receptor synapse. So Frederick then blocked each of the 7 receptors one at a time. And for 6 of them, blocking them had no effect on the capacity of the CS stimulation to elicit the conditioned pause. But when he blocked the mGlu R7 metabotropic receptors. Some of the receptors are ionotropic and some are metabotropic. Bingo, he could completely block the elicitation of the conditioned pause by parallel fiber stimulation. And finally, he went on to show that he tried blocking various channels that might in the axon hill membrane that might be crucial in actually causing the conditioned pause. And he found that there's some weird inward rectifying potassium channel, which when he blocked it, he mostly blocked the elicitation of the conditioned pause. All right. So because some people seem, there's huge resistance within the field to thinking about the implications of what Johansson found. Just to make sure, I'm sure for most of you, the logic of this is pretty obvious, but look, the whole point of obtaining control of the parallel fiber input is that now for the first time, one knows what's in the input, right? And you know that the input signal doesn't contain any information about the CSUS interval. You know it because you're controlling, he was controlling that input. But you can see that by the time you get to his recording electrode, that information is present in what the cell is doing, okay? So the information was not present in the transmitter that was released onto these synapses, but it was present by the time we got to the recording electrode where, well, okay, so where is that information? Well, it's got to be in here someplace, right? Now, when I say it isn't in the synapse, I mean, it's not in the synapse as traditionally understood, which is something that only amplifies or de-amplifies the input signal and maybe prolongs it. Because of course that transformation, I mean, a scalar, applying a signed scalar to the input here is not going to produce, that doesn't contain the information about the duration of the pause. I say that because in thinking about it, if I had to guess where it is, the molecular realization of where the information is stored is probably quite close to the input synapses, because among other things, they've shown that the same Purkinje cell can learn more than one CSUS interval at the same time. Right? In fact, you can elicit two pauses if you make the two very different intervals, one 150 milliseconds and one 400 milliseconds. Then when you stimulate, you get one pause for 150 milliseconds, followed by a recovery, followed by a second pause that terminates at 450 milliseconds. All right, so here's the opportunity. This is Ariadne's thread. You may recall that Ariadne provided Theseus with this ball of yarn or thread that he needed to keep track of where the hell he was in the labyrinth when he was looking for the Minotaur. And of course, when you get into intracellular biochemistry, as you know better than I, it's a labyrinth. There's a hell of a lot going on in there. So one needs a thread. Frederick has handed the molecular biological community the thread. And he says, look, I know the metabotropic receptor. You spritz some glutamate on this receptor, and it's a metabotropic receptor, so it's driving an intracellular communication system, right? A cascade of some kind. It almost, in all likelihood, it's connected to a G protein immediately on the other side of the of the synapse, and the G proteins, they do their thing, and then there's the reason I didn't go into biochemistry is I could never remember the names of all the things in these cascades. Like in the isomerization of opsin, you've got 6 multi-syllable players before you get to the rod outer membrane. Anyway, somewhere in that cascade, and he identified what looks like the other end of the thread, the exit end of the thread with this potassium channel. So somewhere in that cascade is the engram. And the thread leads to the engram, and then it leads on from it. So that gives us a strategy for finding the engram and recognizing when we found it. Before I left UCLA, I participated in a workshop organized by Silva and I think Fanslow on the question was, how would we know that we found the engram if we did find it? I'm here to tell you that the bottom line from that workshop was that most people had no idea. They had no strategy for recognizing the engram if they happened to stumble on it. So first of all, you can follow this thing from both ends, right? That is, we know that until you get to the engram, the biochemistry will not reflect the duration of the CS-US interval. That is, how long the processes last and so on will not be informed by the engram. But we know on the other side of the engram in this cascade, the processes will, the closure of that potassium channel, that's just the final step in this process. And all the steps between the engram and that closure are going to be like the closure itself. They're going to reflect the fact that the information about the CS-US interval has been accessed. It's now in the signal. So I lie awake at night and think, damn, if only I was a molecular biologist. Well, they know how to follow these cascades. I don't mean it's easy, but unless I don't understand, the biochemists and the molecular biologists have already unraveled several of these cascades. And the tools keep getting better and better. And so they can follow it from both ends. And eventually they're going to get to it's a little bit like gene walking, right? You come down the chromosome from one end, you come down from the other end, and eventually you get to a segment that says, all right, somewhere in this little piece is our gene. So then how do you know whether you found it? Well, first of all, the structure of the engram has to encode the duration of that interval. So there has to be an invertible, that is 1-1 mapping, between the duration of the interval, which you control experimentally, and something about the structure of the material that contains the engram. So first of all, if you find such a material, you can already be pretty sure that you're on the right track, that you've really found it. And how can you be sure? Well, once you know what you think the structural modification is, with the tools of modern molecular biology, you can surely produce that modification artificially, insert it into a Purkinje cell in the right place in that cascade, and show that, lo and behold, now the Purkinje cell thinks it was trained with something different, with a different interval. You find that, you'll be on your way to Stockholm. Anyway, so that's the opportunity. Now let me say a little bit more, because when you think about this, you think, Jesus, okay, so that might be a strategy for following the engram, but how can we there's going to be a hell of a lot more to the molecular biology here, right? Yeah, I mean, of course, that's the point. just like there, just like there's a hell of a lot more to molecular biology than DNA, right? But we're beginning to see what other pieces of the puzzle might look like, and they're all looking molecular. And the reason I stress this is that the whole neuroscience community is thinking at the circuit level, right? They don't think molecular biology has anything to do with learning, except insofar as some molecular mechanism preserves the change in the synaptic conductance, right? But once you start to say, no, it's at the molecular level that the information is actually stored. Right? That's where the engram itself is molecular. Well, then there's a question, okay, how do you get from the synaptic inputs to the structural change that is the engram? And how do you get from that structural change back out to firing alterations? Because the engram has to change the firing, otherwise there's no point. There's a Thornquist who did his PhD in either an MIT or Harvard. So some of you probably know him. Anyway, he's discovered, he's now in Gabby Maimon's lab at the Rockefeller, and he's identified a biochemical cascade that's tunable. That is, you can fiddle with it to cause it to time very different intervals. And this is in Drosophila, and shown that it controls, that this cascade is what controls the timing of, I forget. I heard him talk about this and I forgot something about Drosophila behavior. So I've been studying the behavior and neurobiology of timing for more than 20 years. And I'm here to tell you that most of the neurobiological literature is nonsense. This is the first really persuasive story about the material basis of timing that I've encountered. These papers bear on what the read process, this bears on the question, well, how could some structural change that encoded the information affect the molecular biology so that that change was translated into a pause of the requisite duration? Now, perhaps an even nastier question, a harder question to think about is, what does the right operation look like? What the hell goes on between the time that elapses between these two different synaptic inputs and the production of a structural change that encodes the duration of that interval? So some of the more physically oriented people thinking about this kind of process, like Levy and or Lazar, both of whom has physics and engineering backgrounds, of course, are focusing on the fact that what really represents what the information in a signal is really carried by the inner spike intervals. That itself is a controversial issue within neuroscience. Most neuroscience modeling assumes a population rate code, and that assumption says the intervals don't matter. And they do that for mathematical convenience because it allows them to treat the input as a single real valued variable. But if you think about it, that assumption is crazy. And Levy and Lazar are both guys that have been thinking about it. Okay, so as Levy points out, the real driving thing is the charge accumulation at the synapse during two spike intervals. Well, that's of course and that this charge accumulation is the symbol for a latent variable of some kind. That is what it is that the input is giving you information about. And look, if you think I'm telling a coherent story here, I'm not, right? But it at least, to me, says, okay, if you want to start thinking about what might be going on physically, this is a place to think about it. And then, of course, another huge part of it is what the hell is going on with the complex spike, right? That reverberation is really weird. I don't know if there are any other neurons that show that same thing, but it strongly suggests that something pretty fancy is going on in response to that synaptic input. All right, so that's my pitch. I hope now I'm more than open to questions.
Thank you so much. Yeah, fascinating. Folks, anybody have questions?
Yeah, I would have one. So that was super interesting, but I'm not sure if I understand you correctly. So for me, an engram was always something which was, distributed throughout the brain, like several neurons are connected. And then if you find an engram, you find the connection between the different neurons which code for the memory, right?
That's exactly what I asked you to erase from your brain at the start of my talk.
Yeah, I'm in the process.
If you think about it, that theory doesn't ever address the most obvious fact about memory, which is that it's full of facts. Quantitative facts, right? Try it out. Try someone, you won't have any trouble finding someone who is a big champion in that theory. Ask them how you put a number in a synapse, and then watch the hands wave, right?
Yeah, so if I get you right, you instead of looking for the engram between neurons, you're looking for the engram in a single cell, right? In a single neuron. Which and a couple of weeks ago we had David Glansman giving a talk.
Yes, of course.
Yeah, I think he also supports this idea because from an evolutionary perspective, it would mean that memory only occurred when brains evolved, and before that we wouldn't have any memories, right?
That memory only occurred when.
So if the engram would be like a more global thing between neurons, that would mean that memory only evolved at the moment when complex brains have evolved.
Oh yes, right. Of course, yes, right. Whereas on this story, memory may have been there in the Precambrian, right, in the slime. One of the reasons I like Mike is he's open to that idea.
So I'm wondering what the implications of this are for, as opposed to finding engrams in mice or whatever. What about going from the other direction? What about the implications for engineering a simple memory system? So let's say we were going to start from scratch, right, and bioengineer a synthetic construct in a cell. It doesn't have to be a neuron that would implement this whole scheme. I mean, might that not be another way to get at this? Basically, show that it's sufficient, right? So implement that in the cell using molecular biology tools, and then show that that's sufficient to answer the properties of associative learning.
Well, I think that's basically a sociological question. That is, in some sense, we know that in totally transparent, unarguable sense, we know that information facts can be stored in polynucleotides, right? One time when I was the person who introduced me when I gave some version of this talk a few years back, in an introduction, showed a video, the frames of which had been run through DNA. I mean, each frame is just a big bunch of numbers, right? And they wrote the frames into DNA, and then they read it out all using molecular biology and converted it back into pixels, right? It was a low resolution short movie of a horse running, right? Shades of Muybridge. Okay, so there's no doubt that that kind of molecular biological story can be told. The question is it the right story? Right? And I would add to that that people like Levy and myself and Oral and Lawton and Sterling, people have begun to think about the biophysics and in particular the energy use. And when you start thinking about the kind of story that my first questioner was, neural net story, the energy use being implied by those models is crazy. So because every spike requires 10 to the 8th ATPs to be hydrolyzed in order to restore the charge, right? Whereas to add one nucleotide, which is 2 bits to a polynucleotide chain, takes 1 ATP. Whoa, that's magnitude difference in the energy requirements for different proposals about the thing, right? To me and to some physicists, that looks like a very powerful argument for where a sensible engineer would locate the memory, right? He wouldn't locate it in a recirculating spike train. I mean, that way lies, the brain would boil over in 10 seconds, right? Just because of the second law of thermodynamics, right? So it seems to me, frankly, that there are very compelling arguments that it must be a molecular story. So then once you think that, then the question is, okay, now we know whose court the ball is in. It's in, that's why I'm talking to your lab. If this is right, it's in your court, and you've got the tools to whack it. The electrophysiologists are never gonna, they don't have the tools, let alone the fact that they're all barking up the wrong tree.
Anybody else, Pi?
Yeah, I had a quick question. So if the engram is in the molecular mechanisms within the cell, then theoretically any cell should be able to encode memory at least to some levels. So is there something? Okay. Including
the sensory cells and the motor cells, right? So on this story, so one of the mysteries is how the brain reacts incredibly fast, right? If you, in some of these things, and it accesses what we know is learned information, and it appears in the behavior on a very short time scale, right? And so how could that possibly be? Well, once the brain has figured out something, it can ship the required information out to the first order sensory neurons. And it can put the information about the required response in the motor neurons themselves, right? So then, in a really efficient organization, you try to minimize the amount of signal flow through the upper levels, right? But how do you do that when the signal flow requires learned information that required very complicated analyses, right? Well, if you put the information in the molecular form, you can put it anywhere. And yeah, in this story, the brain has the structure of the internet, right? All the computation, all the memory and the computation is going on in the servers, right? So how come there's so much traffic on the internet? Well, that's the servers talking to each other, right? I mean, because they're sharing their memory, right? That's why I put my data in the cloud, because it's in Australia and China and Boston and so on. I can probably get it back.
I have a question. Thank you so much for the interesting talk. So given the basic idea that conditioning alters the structure of the cell itself. One prediction that it makes is that it could change the behavior to future inputs, like whatever be the inputs, including the US. So it's not just the CS. So even if you inject the US after the conditioning, the behavior will be different. Would you agree with that prediction? Because if that is true, then it feels a little bit dramatic, right? Because it permanently alters the behavior to all the inputs post conditioning.
Well, I'm not completely sure that I've correctly understood your question, but you judge. Let me see whether the answer looks like it speaks to the question, right?
Mm-hmm.
So you are completely right that a fact has all kinds of implications. That is, learning a single fact can alter many different behaviors in many different ways.
Mm-hmm.
That is God's own truth, and it's a very important fact. So if that's what your sort of preface to your question was, absolutely. So this change should be reflected in other behaviors. Now, we know that that's true at the behavioral level. That is, we know in Pavlovian conditioning that the animal can do surprisingly sophisticated inferences from the different quantitative facts that it has been taught. Right. So let me give you an example from Ralph Miller's work. Suppose you teach the rat that a shock precedes the tone at 1 second. So you have a week during which, or a day, during which every now and then you shock the rat, and 1 second later a tone comes on, lasts 5 seconds. Okay. Then next week, you teach the rat that a light predicts the tone at a latency of 20 seconds. Okay. Just the tone, no shock. Now you test the animal in various ways for how it feels about both the tone and the light. The animal likes the tone, which, if you think about it, it's at the all clear, right? It's the bombing is over, right? The animal likes the tone, but it's afraid of the light. Okay, so why is it afraid of the light? Well, think about it, right? The light predicts the tone, and at least sometimes the shock precedes the tone, right? So the light predicts the shock. Now, Miller has run that experiment 10 different ways. It doesn't matter the order in which you teach him the two things and so on. The animal always does the subtraction, right? And the addition, right? It uses those two quantitative facts to figure out to infer another quantitative fact, and that changes its behavior to the light. Okay, so yeah, this is absolutely the way memory works. And if memory is at the molecular level inside the cell, that's the way. Remember, we know the Purkinje cell can learn more than one interval. Actually, it probably learns thousands. But we know for sure that it can learn more than one interval. And in fact, this gets, given what we know about the behavioral level, this cell wouldn't show the learning that it does show unless it was also learning the US interval, right? Because Pavlovian conditioning, it actually depends on the ratio between the CS-US interval and the US-US interval. Because if you think about it, the weaker that ratio, the poorer the prediction. It's only when that ratio is, it's only when the CS shortens the expected interval quite appreciably that it's a good predictor, right? And that's exactly the way Pavlovian conditioning actually works. And since you can Pavlovianly condition this cell, you can throw it. Frederick, by the way, has gotten, this is a little complicated story, but basically he's shown the same phenomenon in the dish, right? In a slice preparation, right? Okay, so you can condition this neuron in a slice. That means a hell of a lot of complicated stuff is going on inside that neuron, right?
Interesting. Very interesting.
Excellent. Okay. Any other questions?
Yeah, I just wanted to ask quickly. You mentioned, I think, that the eye blink conditioning is one of the only cases where we have a clean course, or we have a demonstrated clean correspondence between behavior and cellular representation. So I just, that seemed to indicate to me that there might be not a single engram, but multiple engrams, or one that we've cleanly identified here. So I just wanted to ask whether we knew anything about whether there was a singular engram or multiple, or perhaps some that are subcellular and some that are intercellular.
No, we don't know anything. That is, there's huge disagreement in the field about exactly that issue. So the simple answer is no. Now, you might imagine I thought about that question quite a lot. And I think if you think about it long enough, you'll realize that there's just going to be one engram mechanism. For the same reason that there's only one gene mechanism, right? I mean, this is as fundamental as it gets. And Mike was talking about, well, what if you engineered a system? Well, if you're going to engineer a computer, the very first thing you have to think about is, well, how are we going to do the memory? Because it's the most important part, right? It has to, you have to be able to put a huge number of facts in there. Storing them has to require very little energy. They have to last more or less indefinitely, and so on, right? You go back and read the early history of computer engineering. Turing was calculating, what if we stored the energy in the vacuum tubes? You know, we used vacuum tube flip-flops. He calculated that 1 megabyte would require the power supply of the city of London. So, these were, of course, these are pre-transistor days, right? So, Houston, we got a problem. Because, I mean, Turing could not have dreamed that I now have something the size of a small stick of chewing gum that holds 40 gigabytes of information. But he knew that having something like that was incredibly important, right? Okay, so the memory is the most important part of the thing, right? It's the key to everything else. And what I'm arguing, it's also the low-hanging fruit. It's going to be much easier to find the engram than it is to figure out all these other questions, you know, like how is it written to, how is it read, and so on. And again, the history of molecular biology tells us exactly that, right? DNA was the easy part. You can explain it in 15 minutes to an intelligent high school student. If you want to get into Evo Devo, right? How the DNA actually produces an organism. Oh, well, now we're at the graduate seminar level and still pretty confused, right? Okay, so the low-hanging fruit is how the information is stored, because it's, from an engineering standpoint, the most important component of the whole system. And you can't really think about all the other mechanisms until you know what that one is, right? I mean, we didn't really know how to think about development until we knew about DNA. So it's the same story here. That's why I claim it's so revolutionary for neuroscience. It'll change the game in ways that you could hardly imagine. So I think there's only one engram, right? Because when it comes to really important mechanisms, there aren't 100 ways of doing it, right? There are a very few ways. I mean, look at phototransduction, right? Okay, there are 2 ways, 3 ways maybe. And so there's the arthropod cascade and there's the vertebrate cascade, but they're both cascades. They both have an opsin at the front end. You know, it wouldn't surprise me at all if there are some interesting variations on the basic mechanism, right? That seems to me, in fact, extremely likely. But that there are biochemically unrelated engrams, well, we won't know that in my lifetime. If we're lucky, we'll know it in your lifetime. For what it's worth, I'll put down a marker on the answer being no. All right.
Excellent. Okay, thank you so much. I think people might be emailing you afterwards as well with more questions.
Fantastic. Thank you, Mike,
Thank
for
you.
the opportunity.